I extend a warm welcome for this international webinar on role of chemists in discovery of drug and vaccine for COVID-19. It gives me immense pleasure to introduce today's speaker, Dr. Mahender Deval sir, Head of Research, founding member of Expansion Technologies, Cambridge, United States of America. He has done his graduation and post-graduation from Usmania University, Ph.D. from Organic Chemistry from the University of South Carolina. At present, Sir is head and principal scientist of Expansion Technologies, which explore the applications of expansion microscopy technology for early stage diagnosis of various cancers, renal diseases, and skin diseases in pathological setup. He is also a postdoctoral fellow in medicinal chemistry from Wayne State University. Sir has also completed various research projects, and he has more than 20 international journals in publications in reputed journals. He has also 4 patents to his credit. He has presented posters in various international symposiums and also conducted various international symposiums. He is also a peer reviewer of international journals like Tetrahedron, Springer, Elsevier, Relay Science, etc. He has conducted many international workshops. So in this context, Sir is aptly chosen for this international webinar as a resource person. So welcome, Sir, and I extend our sincere thanks for accepting our invitation for this webinar. Thank you, Sir.
Thank you very much, Jayanti ma'am, for introducing Dr. Mahender Deval sir. Now, I invite Mahender Deval sir to deliver his lecture.
Good morning, everyone. Thank you so much for a nice introduction, Dr. Jayanti Garu. It's a great pleasure. In fact, it's an honor for me to deliver this talk today in the memory of our beloved ex-Prime Minister, Sri P.V. Narasimha Rao Garu. Thank you so much, Dr. Sarasija Garu, for the invitation. I also would like to thank the faculty and students of Chemistry Department of Sathavahana University for inviting me to deliver this talk. Respected Chief Guest, Sri Anjaneyulu Garu, Guest of Honor, Professor Bharat Garu, and special guest, Professor Manohar Garu, and all the attendees. Thank you so very much for gracing this event with your presence. I appreciate it. Thank you. Now I'm going to share my presentation, then I will start talking about it. Bear with me for 1 second. Can everyone see my presentation? Just say yes to someone.
Yes, sir.
Okay, thank you. So the title of today's talk is, my talk is COVID-19, a pandemic of coronavirus, progress in therapeutic and vaccine developments. In first half of the talk, I'm going to discuss about coronavirus evolution and infection mechanisms, and also I'm going to talk about the components of immune system, how they fight with the virus infections. In the second half, I'm going to discuss about therapeutic and vaccine developments, various different methods that are used to develop vaccines or therapeutic drugs, and also I'm going to give a few examples what the stage they are and progress in their development as well. COVID-19, 5-letter, 2-number word, has been shaking the world from past 8 months. What is this COVID-19? COVID-19 is an infectious disease caused by most recently discovered novel coronavirus. The symptoms, it starts with most common symptoms such as fever, dry cough, and tiredness, cold, etc. As time passes, new symptoms of these coronavirus infections are discovered, such as losing smell and taste. As it gets more and more severe, it may cause pneumonia, severe acute respiratory syndrome, SARS. That's why this virus is also called SARS coronavirus, which causes organ failure such as kidney and lung, ultimately leading to the death. When I said novel coronavirus, why this is called novel coronavirus? Is there any other coronavirus are existing in the world? Let's take a look at the history a little bit. Coronaviruses, one second. Yeah, the human coronaviruses are described in 1960s. Here I have listed 4 serotypes of human coronaviruses that are discovered in 1960s and early 2000s. All these coronaviruses are originated from either bats or rodents, but they are transmitted to the humans via intermediate hosts, such as camels or cows. This first generation of these 4 serotypes of coronaviruses cause very mild symptoms in infected patients, and this is considered to be not highly pathogenic virus. It only targets the immunocompromised patients. However, in November 2002, new coronavirus broke out in Fujian, China, and the SARS coronavirus 1. The source for this SARS coronavirus 1 is also bats, but it transmits to humans through intermediate host civet cats or raccoon dogs. And this infection spread out only 5 continents or 30 countries. That's why World Health Organization announced it as an epidemic, not a pandemic. And the infections may mild to severe infections may cause from this coronavirus 1 infections, SARS coronavirus 1. Approximately 8,000 people from these 30 countries were infected, and 800 of them have declared dead. Also, in 2012, another coronavirus, Middle East Respiratory Syndrome coronavirus, broke out in Saudi Arabia. This was also spread only 27 countries. Again, this was also announced by World Health Organization as an epidemic, not a pandemic. Source of this coronavirus is also bats, but intermediate host is different, dromedary or Somali camel. This coronavirus infection also may cause mild to severe symptoms, which may also lead to the death. The disease caused by this MERS coronavirus is called acute respiratory distress syndrome, ARDS. And approximately 2,000 people from these 27 countries were impacted, and 700 of them were declared dead. As most of you already know that SARS coronavirus 2 broke out in 2019 December from Wuhan, China. And it rapidly spread all over the world and announced by World Health Organization as a global pandemic by March 2020. The source for this SARS coronavirus 2 is also bats, but intermediate host is pangolins. This infections from this coronavirus also cause mild to severe symptoms, which may lead to the deaths. As of today, there were 21.3 million people from all over the world are infected by SARS coronavirus 2. Among those, 767,000 people are died, having United States first in the infections, and India is in third. Okay, when I explained all these 7 different types of coronaviruses, the first 4 are not that infectious, very mild, but last 3 are much more pathogenic and as infectious as current coronavirus. Let's take a look at the similarities and differences of these coronavirus. So, one of the most important similarity of these 3 coronaviruses, SARS-1, MERS, and SARS-2, is the genetic information. Genome, 90 to 95% of the genome matches with bat coronavirus. That's why these are all evolved from bats. And the 80% of the genomic information of SARS coronavirus 2 matches with SARS coronavirus 1. Like these 2 are the brothers. Only 50% of the genome information of SARS coronavirus 2 matches with MERS coronavirus. Like these 2 are the cousins. And another important difference between SARS and MERS coronaviruses is the receptor that they attach to enter into the cell, human cell. For SARS coronavirus 2, SARS-1 is ACE2, angiotensin converting enzyme 2. infectious, but SARS-2 is much more rapidly spreading compared to SARS-1 and MERS. Why? What could be the reasons? Let's take a look at some of the reasons here. Primary spread of this coronavirus to SARS coronavirus 2 is from person to person, coughing, sneezing, or speaking, the respiratory droplets, and also aerosols. And it also has another reason, which is secondary spread, contaminated objects or surfaces. If you look at the table here, I have listed the stability of SARS coronavirus 2 on various surfaces, at various temperatures. At 4 degrees centigrade, which means in the fridge, the SARS coronavirus is active up to 2 weeks. At 37 degrees centigrade, approximately the body temperature, not inside the body, outside the body, it is active up to 2 days. At 70 degrees centigrade, which means almost boiling water, it is cleared out within 5 minutes. And another interesting feature is, if you look at the regular paper and tissue paper, it is active up to 3 hours. But currency note and a cardboard, these two are also made of papers, but it can survive. It is stable until up to 4 days on currency note and up to 2 days on cardboard. And depending upon the other materials, cloth, wood, glass, plastic, it can be active up to a week also at 22 degrees centigrade, which is a room temperature. And another reason is long incubation period, approximately 2 weeks of incubation period, up to at least 1 week or 5 days. The symptoms may not be shown up, but the virus will be growing inside the body, infected person's body, which can be easily spread out. And recently found another important reason for the rapid spread is mutations. This one single particular mutations, aspartic acid D, at 614th position of spike protein, mutated into a glycine amino acid. Here, this is a cryo-electron microscopy structure of the spike protein of coronavirus, and it has 2 subunits, S1 and S2 subunits. The mutation happens into this particular region where these 2 subunits are joined. And this is a zoomed region of here. This is the amino acid, aspartic acid, which is present in the in the wild type coronavirus. But mutated coronavirus has the glycine amino acid. If you look at this data here, from January, since the virus infection started, the genome sequencing was done on the viruses that collected from the patients. In January, there is no mutation. Only aspartic acid was present in the spike protein of the viruses that was detected in the patients. In February, also 0% mutations, but it started mutating in March and also spreading fast from March onwards as well. 26% of the virus has glycine mutation. In April, it went up to 65. In May, it up to 70% of mutation. What this mutation does is it gives much more stability to the spike protein, and more protein is incorporated into the virus particle so that it can interact with the human cells and infect much rapidly and much faster. So what is this coronavirus? Let's take a look into the much more deeper look into the parts of the coronavirus. Once Peter Madower, he's an immunologist and a Nobel laureate, defined virus as simply a piece of bad news wrapped up in proteins. Exactly. Like living cells, viruses contain genome. It could be RNA, it could be DNA. In coronavirus case, it is a large single-stranded positive RNA. Large RNA is securely encapsulated by nucleocapsid protein. If you look at this and also another unique property of this virus is this RNA genome is very well securely spirally wrapped up by nucleocapsid protein. This is a unique feature for the coronaviruses only. Not other RNA viruses have this kind of feature. It also has a very strong, well-developed structural proteins like nucleocapsid protein, membrane protein, envelope protein, and spike protein. Spike protein is also a unique feature of these coronaviruses, not other RNA-viruses. Coronavirus uses this spike protein to enter into the human cell to replicate more and more. Unlike living cells, the viruses in general does not divide into 1 to 2, 2 to 4, or 4 to 16 like that. It is completely different. Mechanism is completely different, and it requires a host cell to be active and replicate itself. This coronavirus belongs to zoonotic virus family. That means it exists in the animals, but it infects animals to animals, animals to humans, humans to humans. When I say this spiral genome uniqueness of the spiral genome, let's take a look into the much more details of the genome. As I mentioned, it has a large genome with approximately 30 kilo base pairs of nucleotides that encoding 29 proteins. This is a genomic map in 2D map, which may be divided into two parts. First part has two open reading frames that encodes 16 non-structural proteins. And second part has 4 structural proteins and 9 accessory proteins. What do they do? What are the functions of these proteins? So among all the 16 non-structural proteins, only few of them are very well characterized. Some of them are still, we do not know what their functions are. Among those, 2 proteases, NSP3, NSP5, are very well characterized. And another enzyme is RNA-dependent RNA polymerase, NSP12, which is called replicase. This is also another unique enzyme that only coronaviruses have. What it does is it transcribes the positive sense RNA into a negative sense RNA so that the cellular enzymes can make more and more of that RNA. Also, it takes the help from NSP7, NSP8. NSP1 also has recently discovered that it suppresses the immune response from the human body. I will explain you that in my upcoming slides. The functions of accessory proteins are not clearly known, but the structural proteins have very unique functions. Nucleocapsid protein provides the security to the genome, very well nicely wrapped in it. And spike protein is very important to enter into the cell. Envelope and membrane protein together, they coordinate to give a stable structure to the virus. Now, let's take a look at the cell entry mechanism. So, this is the cell membrane of the human cell. As I mentioned in my previous slides, the spike protein attaches to the ACE2 receptor on the human cell. Once it attaches, an endosome will be formed. And once the endosome is formed, the spike protein fuses into the viral, cellular membrane and releases the RNA from the virus into the cell. Entire this process is called endocytosis. So here, if you look at this cartoon, which clearly shows the interactions between the spike protein of virus and ACE2 receptor of a human cell. The spike protein, this is the 2D map, which has 2 subunits. One is S1 subunit, this part here, and S2 subunit. In this huge protein, this particular small region, which is called receptor binding domain, plays a very important role in attaching to the ACE2 receptor. Once the virus releases the RNA into the human cell, immediately human cell enzymes, what they do is they translate the open reading frames into functional non-structural proteins, the proteases and replicase. Once the replicase is made, it started transcribing the positive sense RNA into a negative sense RNA. And once again, the cellular enzymes transcribes these negative sense RNA into much more and more RNA, then makes the components like structural protein RNA and accessory protein RNAs, which will be translated by, again, human cell enzymes and transported to their respective places, such as endoplasmic reticulum and Golgi body. Where they mature, they basically refine, and the virus assembly process will take place at the membrane of the Golgi or endoplasmic reticulum. All the structural proteins will start incorporated to the membrane, and RNA comes from the cytoplasm. Then the functional virus will be developed, assembled, and an endosome will be formed. Then exosome will be formed. Then the virus will be released into the outside of the cell. The process is called exocytosis. So when a virus enters into the human body, the first thing that the human body does is activates the immune system. So let's take a look at the immune system. Just 1 second, I think my slides are not showing properly. This Zoom app is giving some trouble. Let me clear this out before I continue my talk. All right. So the human immune system, in humans, we have 2 types of immune systems. One is innate immunity. The second one is adaptive immunity. Every individual has innate immunity by birth. This is a frontline defense which reacts non-specifically, attacks the infection site, goes to the infection site and attacks the virus or any other foreign particle or bacteria, anything comes into the body. This innate immunity will be activated within hours. Maximum it takes only 12 hours. Within like 6 hours, it will be activated to clear out the infections. And adaptive immune system is like a backup defense. It requires antigens. It's a specific defense, a specific response it has based on the antigen molecules. It will be activated slowly, but once it is activated, it will last long time, months to years. And also, adaptive immunity does not come by birth. It has to be developed as the human grows. And that's why, even though the innate immunity and adaptive immunity very well developed in adults, but older generations, with people with more than 60 or 70 years, their functions will be slowed down because of the age-related issues, age-related properties. That's why the older people are much more prone to the infections of this coronavirus too. There is always crosstalk between innate immunity and adaptive immunity to fight with the infections or viruses or bacteria. Innate immunity. So innate immunity composed of various different cells. The first thing what it does, it releases some chemicals which are called cytokines and chemokines. These molecules reaches to the infection site and generate the signals and activates the inflammation mechanisms. Once cytokines activates the inflammation mechanisms, macrophages reaches to the infection site and start eating the virus based on the receptors, cytokine and chemokine receptors that are attached on the virus and virus infected cells. So they start engulf the viruses. Then nature killer cells. These nature killer cells activates the suicidal mechanisms in the virus infected cells, so that the cell will be died itself, a process called apoptosis. While these two cells are doing their job, antigen presenting cells goes to the infection site and collects the antigen molecules such as spike protein or nucleocapsid protein from coronavirus, and then runs towards the adaptive immune system. Even though the innate immune system has various different cell types, I only mentioned here three major cell types. Once antigen presenting cells brings the antigen molecules and gives to the adaptive immune cells, adaptive immune system has two types of cells, B cells and T cells. B cells, once it has the antigen molecules from the antigen presenting cells, it generates various different types of antibodies, including neutralizing antibodies. So neutralizing antibodies, what they do is they basically arrest the virus. They bind to the spike protein. They cover the spike protein surface so that the spike protein will not attach to the receptor molecule to enter into the cell. Once it arrested by these antibodies, the macrophages will clear it out from the human body by eating or engulfing it. T cells generates two types of cells, T helper cells and T killer cells. T killer cells has the same function as the nature killer cells. This is an additional force to the innate immune system, nature killer cells. They lyse the cells, they clear out the infected cells. And T helper cells, they generate the memory cells. These memory cells play a very important role. What do? Is they remember the characteristic features of that particular virus and stay in as a reserved army. And they also remembers how to fight with that particular virus. These are called memory cells, and once they are generated, they last months to years. However, even though the innate immunity and adaptive immunity perfectly works to clear out the virus, but somehow coronavirus escaping it. There are various hypotheses are discovered how this coronavirus escapes from the immune attack. The very first thing what it does is it sends the wrong signals to the immune system so that they release a flood of cytokines, which is called cytokine storm, that activates lots of inflammatory pathways, inflammatory mechanisms, which causes damage to the tissue. The tissue damage causes organ function failure. Once organ failed, multiple organ failure leads to the death. So there are various hypotheses are developed to explain how the SARS coronavirus 2 escapes from the immune system. There is some of the literature suggests that accessory protein may be helping the virus to escape from immune attack. And second hypothesis is the conformational flexibility of the spike protein. This is the cryo-electron microscopy structure of spike protein. This is S1, S2 subunits. This is the RBD, receptor binding domain. So if you look at that, receptor binding domain is in a standing position, which is an active form of the spike protein. But the second form, it has a kind of a sleeping position. This is inactive form. So when immune system is searching for the virus antigen, it goes down into this inactive form so that it escapes from the immune system attack. The third hypothesis is sugar coating of spike protein. Spike protein is decorated with N-glycans. N-glycans are nothing but multiple sugar molecules forms a glycan which attaches to the surface of the proteins that gives stability to the proteins. So spike protein has approximately 24 to 27 N-glycan sites, which completely covers the spike protein so that it will be escaped from the immune attack. Recently, last week, a study was found that NSP1, non-structural protein 1, binds to the ribosome tunnel where mRNA is translated into the protein. So it inhibits the mRNA translation of immune system proteins. That's how it's blocking the to generate the immune components and escaping from the immune system attack. If you look at this is the tunnel where mRNA, orange color mRNA is being translated, but this purple color NSP1 binds right in the middle of the tunnel so that RNA cannot translate and release into the endoplasmic reticulum or in the bloodstream. So when the virus takes over the immune therapy, immune system, we need additional force, additional material to support the immune system. That's where vaccine and therapeutic development comes into the place. So to protect from the viruses, we have two options. One is vaccine. Vaccine, it only prevents, it's a preventive measure. It can prevent the virus infection, but it does not cure the disease that caused by the virus. And to cure the disease caused by a virus, we need treatment options like antiviral therapies, antiviral agents, antiviral drugs. So first, I'm going to discuss about the vaccine. What is a vaccine? How it works in the human body? Then what are the stages, development stages of a vaccine or a drug molecule? Then I will go into various different methods of vaccine development and with an examples I will give. So vaccine is nothing but, it could be a virus, a part of a virus, or a protein of a virus, or RNA or nucleic acid of a virus that will be injected as a vaccine into the human body to initiate the immune response. Basically, the vaccine strengthens of the adaptive immune system. As soon as it is injected, the antigen presenting cells collects the antigen molecule from that vaccine candidate and reaches to the adaptive immune system to generate the memory cells and neutralizing antibodies. The memory cells, as I mentioned, they stay in the body for months to years to fight with the infection. When the real virus comes into the body, the memory cells will be activated and neutralizing antibodies will be activated and reaches to the infection site and clears them out quickly and soon. That's why vaccines are kind of preventive measures. So there are two stages in either vaccine or a drug molecule development: preclinical stage and clinical stage. In the preclinical stage, the drug or the vaccine is developed in the lab and first tested in the human cells using the pseudoviruses, then tested in the animals for safety and efficacy. Once it passes the stage, it probably a couple of healthy individuals can also be tested in the preclinical trial. Once it passes the preclinical stage, it moves into the clinical stage. Clinical stage has 4 different phases: phase 1, phase 2, phase 3, and phase 4. In phase 1, approximately 20 to 80 volunteers will be given the vaccine or drug to test safety and various different doses. And approximately 70% of the candidates passes phase 1 stage, phase 1 to go to the phase 2 stage. In phase 2 stage, more than 100 people will be tested for efficacy or side effects of that particular vaccine candidate. Approximately 33% of the candidates that reach into phase 2 will be moved on to the phase 3. And phase 1 and 2 will be only tested healthy adults, healthy individuals between the age groups of 18 to 55 only. When this candidate, a potential candidate, reaches to the phase 3, where thousands of people will be tested for the safety, efficacy, and if there is any adverse reactions. And in this stage, not only the healthy individuals between 18 to 55 age group, but also more than 55 age group people will be tested. Also, people with pre-existing conditions such as diabetes patients, dialysis patients, and heart patients, and so on, various different patients will be tested for safety, efficacy, and adverse reactions. Once this stage is passed, the regulatory committees will decide whether this can be released into the market, to the real world, as an investigational new drug or new medicine to follow up the long-term side effects. Well, only less than 10% of the candidates will pass this stage. So this entire process takes approximately 10 to 12, 13 years because preclinical stage itself takes about 5 years. However, current situation, because of this COVID-19 pandemic, the regulator authorities are developed some fast-track methods so that these phases can be in parallel, simultaneously conducted or parallelly run. So to develop a vaccine, one important thing is antigen molecules are very important from that particular virus. So in case of coronavirus, the structural proteins, spike, envelope, matrix, and nucleocapsid proteins play a very important role to as an antigen molecules to develop the vaccines. Mostly the spike protein or nucleocapsid proteins are being used at the moment. There are various different methods to develop the vaccines. Viruses can be used as a vaccine, nucleic acid-based vaccines, viral vector-based vaccines, or protein-based vaccines are approaches are there to develop the vaccines currently. As of today, according to World Health Organization, approximately 167 total potential candidates are under development from all over the world from various different countries, either independently or in collaboration. 29 of them are already in the human clinical trials. So now the first approach is using virus-based approach, developing the vaccines. In this approach, there are two different methods. Using live attenuated virus. Attenuated means weakened virus. The virus as it is will be taken, it will be passaged in the human cells or in the animals to multiple generations until it becomes weak and weak. So that will be utilized as a vaccine because it will activate, induce the immune response, but it will not cause the disease. There are multiple examples are already there in like smallpox vaccine, MMR vaccine, chickenpox, influenza vaccines are developed by using this live attenuated virus method. Indian Immunologicals, Serum Institute of India, Codagenics are also trying to develop vaccine for the COVID-19 using this particular method, which are probably in the preclinical to phase 1 stage. The second method is using inactivated virus. So virus will be inactivated by chemicals such as formaldehyde and also heat. Heat-induced inactivation of the virus is also methods are all there. And using the albumin also used to inactivate the virus. So the virus will be basically killed. Virus particles will be used as a bait, as a trick to get the immune response. Examples in this method, examples of the vaccines developed using this method are poliovirus, hepatitis A, and rabies. Chinese companies Sinovac and Dynavax are working on developing a vaccine using this inactivated virus method, and also Bharat Biotech from India. They are also developing an inactivated virus vaccine to fight with COVID-19. Both the companies are presently conducting phase 1-2 clinical trials. They successfully completed their preclinical stage and conducting the phase 1 and 2 clinical studies in parallel. The second approach is using viral vector-based vaccines. And there is also two methods available in this approach. Using replicating viral vectors. For example, the weakened virus will be taken, as I mentioned in my previous slide, attenuated virus. That virus will be taken and its genome will be engineered. The entire genome will be removed and incorporated the spike protein or COVID-19 virus genome parts into that attenuated virus and used as a potential vaccine. So this vaccine is a virus. It activates the immune response but does not cause any disease. It replicates like a real virus. The second method is using viral vectors, these non-replicating viral vectors. Adenovirus is the most commonly used virus in this method, which is a cold virus. It doesn't cause severe symptoms in the humans. And again, this viral vectors can be engineered to incorporate the genome of coronavirus parts, such as spike protein or nucleocapsid protein, to induce the immune response. And this non-replicating viral vectors cannot replicate. They cannot make more and more of this virus. They just, whatever you inject, that will create the immune response. So Consino Biologics, a company from Chinese company, first company which successfully completed the preclinical stage by February. The virus detected in December. By February, they completed the preclinical stage and they entered into the phase 1 clinical trials by March. Presently, they are using adenovirus type 5 serotype as a vector, and they're using the entire spike protein genome engineered into this 85 adenovirus vector to develop the vaccine. Presently, they're conducting phase 1-2 clinical trials simultaneously. They also, a good sign is they also released some interim data. Approximately 80% of the participants reported systematic reactions, mild reactions such as pain, fever, fatigue, headache, and muscle pain, and so on, which are resolved within 48 hours. That is a good sign. It's not a bad sign. So it has a good safety profile, and there is no serious adverse events were found when these vaccines tested in the healthy individuals. It induced the immune response within 14 days and retained up to 28 days. This is just interim data from 100 patients, 100 participants. Right now, they are testing this vaccine on 108 in phase 1 and 508 participants in phase 2. Another company, the Oxford University, together with AstraZeneca and BARDA, an American government agency, funding agency, they are also developing a potential vaccine for COVID-19 using a chimpanzee adenovirus. So they engineered the chimpanzee adenovirus using this entire spike protein genome into that, and to develop the potential vaccine for the COVID-19. They have successfully completed their preclinical studies. Presently, they are conducting all 3 phase clinical trials simultaneously. Approximately 1,077 patients in phase 1 and 2, and 12,000 participants in phase 2 and 3. Even there are still more, recruiting more and more people. And they also released interim data from phase 1 clinical trials. When this vaccine is administered 1 to 2 doses, approximately the rapid immune response is induced 91% of the participants, and the memory cells and antibodies that are successfully generated, they are sustained and retained up to 56 days. This vaccine candidate also has a very good safety profile. There is no serious side effect, adverse events were found. There is some transient local systematic reactions were found, like I said, headache, fever, muscle pain, or injection site pain, but these were kind of cleared when paracetamol is used as a prophylactic a day before the vaccine administration. Presently, like I said, they're conducting the 2-3 clinical trials simultaneously in 3 countries, UK, Brazil, and South Africa. The AstraZeneca's goal is to supply approximately around 2 billion doses globally by end of next year. Another company, Janssen, sister company of Johnson & Johnson, also using another adenovirus, which is a 26 serotype of adenovirus vector engineered with the spike protein gene to develop a potential vaccine candidate for COVID-19 infections. They have completed a preclinical stage successfully. Presently, they are conducting the phase 1 and 2 clinical trials, more than 1,000 patients. These trials are already ongoing in USA and Belgium, and they're also planning to conduct in more other countries like Japan, Netherlands, Spain, and Germany. The goal of Johnson & Johnson is to supply more than 1 billion doses globally by end of next year. Second approach is using nucleic acid to develop a potential vaccine for the COVID-19. So this is a 3D model of SARS coronavirus, and this is the cryo-electronic microscopy structure of spike protein, S1 subunit and S2 subunit, and this is the receptor binding domain here. So in this approach, a DNA that either corresponds to entire spike protein or a part of the spike protein, could be S1 subunit or S2 subunit or only receptor binding domain, will be injected straight into the human body when the DNA goes into the body, the enzymes transcribes it into RNA. Then RNA will be translated into the spike protein. When the spike protein is detected by antigen presenting cells, the rapid immune response will be released to generate the memory cells and antibodies. And another approach is using the RNA, same as either RBD, S1, S2 subunit, or entire protein RNA that encoding the protein will be formulated in lipid nanoparticles. RNA cannot be injected straight and direct. It will be formulated in lipid nanoparticles, and when it is injected, the RNA will be released into the body and translated into spike protein. Immune response will be induced. Moderna Therapeutics and National Institute of Health from United States together developing a potential vaccine candidate for COVID-19, which is mRNA 1273. They have their own lipid nanoparticle, exclusive lipid nanoparticle method that they encapsulated the entire spike protein RNA that encoding the spike protein and administered through as an injection into the muscles. They have successfully completed the preclinical studies, and also they have released. They have started their phase 1, 2, 3 trials in April, June, and July simultaneously. 120 participants, 600 participants. Presently, they are conducting a large phase 3 trials in 30,000 patients. That's on 30,000 participants. They also released interim data. When this vaccine is administered, either 1 or 2 doses, induced a rapid response, provided a good safety profile, no serious adverse events, except transient local systematic reactions, which will be cured within 48 hours. And presently, they are conducting large clinical trials. And the goal for the Moderna is to deliver 500 million to 1 billion doses by end of the next year. Another company, BioNTech, together with Pfizer and Fosun Pharmaceuticals from China, are also developing a potential vaccine candidate for COVID-19 using mRNA. So they started with 4 different vaccine candidates. The first one is they used unmodified mRNA that encodes only receptor binding domain of the spike protein. And the second candidate, modified mRNA. The mRNA will be modified using this N-methyl pseudouridine. Usually mRNA contains this uridine base in there, but they modify using the N-methyl pseudouridine which regulates the translation and transcription much better than the original mRNA, the uridine. So in the second candidate, they used only receptor binding domain encoding mRNA with a modified base. The third candidate, BNT161B2, this is also modified RNA, but it encodes the entire spike protein gene. The fourth candidate is self-amplifying mRNA of spike protein. All these 4 candidates successfully completed. Among all these 4 candidates, the middle 2 are successfully completed their preclinical and moved into phase 1. Phase 2 and phase 3 studies are simultaneously in parallel conducted. And a recent report, they also released an interim result with 2 doses, induced rapid immune response. Cells and memory cells and antibodies are retained up to 43 days. But the BNT161B2 candidate is more ahead of than the 161B1. This is in the 3rd phase clinical trials. Currently is ongoing in USA, Argentina, Brazil, and Germany. The goal of Pfizer is to deliver 100 million doses by end of this year and about 1 billion doses by end of next year. Innovia Pharmaceuticals, this is another company developing a vaccine named INO4800. They are using a DNA-based method. They also developed this device, Electra, to administer the vaccine through skin. This is an electroporation device. So they are using a plasmid DNA that encodes entire spike protein. Successfully completed the preclinical studies. Currently, they are conducting phase 1 and phase 2. They started in July, phase 1, August, phase 2. About 160 participants in USA and Korea. And this is the only company has a vaccine candidate in phase 2 clinical trials for MERS coronavirus, the other coronavirus. And Zydus from India also This is the second company other than Bharat Biotech developing a vaccine using the plasmid DNA for the COVID-19 infections. And presently they have successfully completed preclinical studies in June. Presently they are conducting, according to their press release, they are conducting phase 1-2 clinical trials in parallel on 1,000 participants. Not a lot of information is revealed from the company. What kind of a plasmid they are using, how much plasmid, either the spike protein, entire spike protein, or only RBD, or only subunit, not available. But let's hope for the best. Hopefully they may get success. Other approach is using protein-based or protein subunit-based vaccines. So using, in case of coronavirus, using only entire spike protein directly formulated in the lipid nanoparticles or any other formulations and injected into the body to induce the immune response. Maybe only RBD domain can be also be formulated, or S1 subunit like this will be formulated and to induce the administered to induce the immune response. A company called Novavax developing a vaccine candidate using this method, protein subunit method. NVX is the vaccine candidate for this protein-based methods, COV B2373. They have also successfully completed preclinical studies. They started phase 1 in July, phase 2 recently on 131 patients, participants. They also released some interim data, 2 doses, induced immune response, cells and antibodies retained up to 28 days. And there is no severe adverse side reactions, side effects were found. Safety profile was good. Phase 1 and 2 clinical trials are currently underway in Australia. So far, I have discussed about the vaccine development methods and with some examples. Now I'm going to talk about the therapeutic molecules. There are 2 methods to develop the therapeutic molecules for these COVID-19 infections. The first one is repurposing the existing therapeutics. This process is much faster than any other processes. So in this method, the existing broad spectrum of antiviral agents will be screened, tested against COVID-19, coronavirus 2. Also, other drug molecules for heart attack molecules, or BP drug molecules, or any other treatment molecules also screened to find a potential candidate. The second method is developing a new drug targets based on the genomic information. This is a time-consuming process compared to the repurposing. Here, I have listed several candidates that showed a promising result when they were screened against COVID-19. All these molecules are, except remdesivir, are already approved clinically to treat various virus infections. These 2, lopinavir and ritonavir, are 2 approved for HIV infections. These, when they are repurposed against COVID-19, they are inhibiting the protease. They are acting on the protease molecule, protease enzymes of the COVID-19. These 2 molecules are approved for hepatitis B infections. And this molecule is acting on the spike protein, and this one is inhibiting the nucleocapsid protein synthesis. They also showed some promise towards the COVID-19 infections. These 3 molecules, except remdesivir, these 2 are already approved, favipiravir and galidesivir, approved for Ebola virus infections. This was under development to treat the Ebola virus infections. When remdesivir was repurposed against COVID-19 infections, it showed very promising results, and now it is already available in the market as an emergency use. So, favipiravir. This is the chemical structure of the favipiravir. Once it entered, it will be generated an active form, triphosphate form of the active drug molecule, which successfully inhibit the replicase activity, RNA-dependent RNA polymerase of virus. And presently, Glenmark is marketing this, and some other companies are also marketing this drug in India to treat mild to moderate patients with mild to moderate symptoms, because this is approved as a restricted emergency use by Drug Controller General of India. Presently, it is under phase 3 clinical trials are going on from various different companies on this drug molecule. Remdesivir. This molecule also blocks replicase activity. Initially, it is developed by Gilead Life Sciences in the USA, and presently, this was also approved as an emergency use by FDA in USA and DCGI in India for patients with severe symptoms. Cipla and Hetero are marketing, and some other companies are also marketing in India. Gilead is marketing it in the United States. This is the prodrug, structure of the prodrug. Once it is enters into the body, it will be changed into, it will be transferred into the active drug, which mimics one of the nucleotides, adenosine. And presently, Gilead Life Sciences is conducting huge phase 3 clinical trials, approximately in 5,000 patients. And according to their press release, they have already submitted as an approval application as a new drug, investigational new drug. So anytime it can be approved by the regulatory authorities, and it even though it is used now as an emergency use, it can be used as a regular use once it is approved by the regulatory authorities. This is another molecule. Emory University and Ridgeback Bio and Merck together developed this particular molecule EIDD 2801 to treat the Ebola virus infections. This was also under clinical trials for the Ebola infections. This molecule also blocks the replicase activity, and when it is repurposed to against coronavirus, it showed very promising results. And the good thing about this molecule is it is taken as an oral pill, like a tablet. It doesn't have to be like an injection. This is the structure of the active drug. This is the structure of the active drug, which mimics the uridine and other nucleotide of the DNA RNA. Successfully completed the preclinical right now. The phase 1-2 trials are ongoing. Hopefully by end of this year or maybe early next year, this molecule is available to treat all sorts of patients from coronavirus infections. And there are some other therapies also available, such as hyperimmune therapies using the neutralizing antibodies. This is what my lab is working on. Unfortunately, due to the confidentiality and conflict of interest, I may not give you the data, but I'll give you a glimpse of what we're doing. I previously mentioned this is the cryo-electron microscopy structure of the spike protein with S1, S2 subunits and the receptor binding domain. So the neutralizing antibodies, what they do is they bind to the receptor binding domain to inhibit the interaction between the S2 receptor and spike protein. Here, if you look at this yellow antibody, CR3022, generated by isolated from a convergent plasma, bound to the receptor binding domain, but it did not bind where it's supposed to bind to interrupt the interactions between the S2 and RBD. So what we do is we take these type of antibodies and using machine learning algorithms and computational algorithms, make them better binders and synthesize them in the lab and test them on the pseudovirus and then real virus to move forward towards the animals as well. This is what we have been doing. In summary, so far, I have discussed about the evolution of coronavirus towards the COVID-19 pandemic. And I also have discussed about the immune system components, the body surveillance immune system, how it fights with the viral infections, in failure of this immune system, how the disease causes. Then I also have discussed about how to develop the vaccine, how vaccine development process, and some examples, therapeutic development process, and some therapeutic molecules. However, so far, we do not have any vaccine candidate, either SARS coronavirus 2, neither MERS or SARS coronavirus 1. Even SARS coronavirus 1 is there from 18 years. We still don't have any vaccine for the SARS-1 infections to fight with. MERS is there from 2012, almost 8 years. One company or a couple of companies have developed the vaccine, which are in phase 2 clinical trials at the moment. So how to protect ourselves when, since we don't have a vaccine or a proper treatment? So these are the chemistry cartoons. I think the chemistry students understand better. So social staggering, which implements the social distancing. That's one of the important points. This is a ethane molecule, eclipse versus staggered conformations, and the carbon molecules wear the mask. This one didn't wear. This carbon is yelling at the other carbon, where is your mask? Our hydrogens are yelling at each other, stay away from me. Maintain the social staggering. So when the carbon rotates approximately 180 degrees, it forms a staggered conformation intai, and hydrogens are much more far away from each other. So that mentions the importance of the social distancing. And staying home, unless until you have an important work, don't go outside because there is a coronavirus infection is out there. Eat well, sleep well, and be active. These are all chemistry cartoons. The fluorine molecule eats all the electrons, very greedy for the electrons, right? So you eat healthy food. Rest, this is the dopamine molecule, which induces the sleep, good sleep, rest, and then active. IR radiation activates the aromatic molecules. Be active, do some exercise to strengthen up your adaptive immune system. So my journey, I have started from Usmania University campus, Department of Chemistry, where I started learning my chemistry knowledge. Then I have worked a couple of years in IICT India, Hyderabad, where I started my research career. All the wonderful colleagues, I still remember them. Then I moved to Department of Chemistry, University of South Carolina for my PhD, where I successfully completed my PhD in organic chemistry, where I have synthesized various different molecules. Then I moved to Wayne State University for my postdoctoral studies, where I have utilized my organic synthetic skills and learned how to culture the bacteria, how to test the synthesized compounds on the bacteria. Then I moved to MIT, where my first job, where I started working with mammalian cells, human cells, utilizing them techniques that I have learned at South Carolina, University of South Carolina, and Wayne State University. I learned how to culture the mammalian cells, test them on the compounds on the mammalian cells, and I have studied various cancer mechanisms. Presently, I work at Expansion Technologies, where we are developing early stage diagnostic methods for various different cancers. Prostate, breast, pancreas, liver, kidney cancers, where I'm working with human tissue sections here. Right now, we put this project aside, and then as an emergency, we took up the COVID-19 project. I'm synthesizing the antibodies in the lab to develop a hyperimmune therapy for the COVID-19 infections. Having said that, my apologies. I took about 8 minutes over. Thank you so much for your patience, and all my friends, colleagues from various places, my mentors, my professors, my teachers, gurus, everybody from all these various places has helped me to grow, including my family and friends as well. Without their support and help, I don't think I would have been where I am and what I am today. I would like to thank all the my mentors and gurus, everybody, for their support, unconditional support. Having said that, thank you so much for your patience. I'm happy to answer any questions if you have.
I request Jayanti Madam to give formal vote of thanks. Jayanti Ma'am. Sir, there are some questions, sir.
Sure. Please go ahead. Happy to answer.
This is Professor Ashok, Department of
Hello, sir.
University.
Good morning.
Very good morning. Thank you very much for your highly informative and useful talk. I am very much confident that all the participants have very much benefited with your enlightened talk on COVID-19.
My pleasure.
Oh, there are some queries from participants.
Sure.
The Busra Khanam has asked, virus needs a living body to activate itself. Why it is said that it is active at surfaces too for so and so hours or days?
Good question. So
This is the question, sir.
yeah. So virus does not need a human cell to be active. for its replication. To make it more and more, it requires a human cell. And interestingly, this coronavirus is stable on various different surfaces for extended period of times. That means even though it is not replicating itself, it is still active to infect the people. So after that period of time, it will be completely cleared out, right? So it cannot be replicated without a host, but it can still stay active for like an X amount of time. All the viruses, some viruses within minutes, they will be destroyed. Some viruses take hours, some viruses takes days.
Another question, sir.
Sure.
Mr. Srinivas asked, what is dexamethasone and does it work against COVID-19?
Yeah, very good question. So dexamethasone is a steroid. It controls the inflammation, but it does not, there's no studies that says that it inhibits any COVID-19 enzymes or COVID-19. So this is a supportive therapy. So when you control the inflammation, immune system automatically clears out the vaccine. So I don't see there is any evidence that dexamethasone stops the infection or kills the virus or clears the virus.
Another question,
I hope I answered the question.
There is a talk that antibodies produced in the COVID-infected patients will last only for 2 to 3 months.
Yes, some patients displayed. Yeah, this is the thing. This is a 8-month-old virus from now so far. It's every month, every year, it's changing. Sorry, every month some new data is coming out every month. But yeah, these studies, there are some studies says that up to 2-3 months are surviving, but not all the patients. It could be only because of their body style, body nature. That's why again the vaccine development takes a long process. You have to test the durability of the antibodies and the memory cells generated by the vaccine. So that's why at least in my opinion, you have to take at least 1 year. They have to survive up to 1 year, memory cells and antibodies, generated antibodies. Otherwise, within 2 months, 3 months, if they are cleared out, then the vaccine is not potent enough to generate the stronger antibodies or stronger memory cells.
Another question, sir.
Sure, please, sir. Go ahead.
Russia has released a vaccine without conducting third phase of trials.
To be honest,
Advisable to use?
Yeah, to be honest with you, I don't believe in that vaccine at all, the Russian vaccine. Russia has Russia provided great eminent scientists to the world. Enter chemistry, physics, great physicists, great chemists came from Russia. The periodic table was first designed by Mendeleev from Russia. But now I think, no scientist speaks in Russia. They don't release any data. Everything comes out the newspaper or someone else, officials. When a scientist or a doctor speaks about the vaccine, there is a reliability. But other sources, I wouldn't. First of all, my opinion, this is my personal opinion, that I do not believe the Russian vaccine at all. They didn't, how they made it, what they used it, no idea. In my talk, I explained all the information is available in all science publications. Science is reliable when it is shared and discussed transparently. That is the science. That's the real good science. But Russians, it's not a space science. You hide everything and one day, hey, we went to the Mars, we went to the moon or blah, blah. That's okay. Nothing is happening. But vaccine, you're playing with people's life and death, right? So I don't see any transparency in Russian vaccine. I don't believe in it. And then I think I gave you the message. You can interpret from my message.
Another question, sir.
Please.
In normal times, vaccine development takes many years. But today, we are seeing accelerated timelines coupled with pressure from government, the media, and the public. How researchers dealing with this?
Yeah. So it's not a pressure from the government or media or anything. It's a pressure of inside, like the spread, the deaths, everything, right? So even though it is fast-tracked, the clinical trials are being conducted in parallel. They're all taking all the safety measures. Nothing is being neglected. Nothing is being removed from the process. Only thing is the process is going in parallel, and also participants, the volunteers are available to take the vaccine to provide the data, right? So nothing to worry about that. And researchers, any scientist, I don't think he will compromise the quality of the vaccine or drug molecule to release into the market or into the people. That's why, there are various different phases. Even, you can, if it is not past the safety and efficacy profile, the regulatory committees will never approve that at all. Not to worry about that.
One more question asked by Dr. Prashant Babu.
Please.
What type of vaccine released by Russia?
What type of vaccine released by Russia? Is that the question?
There are different types of vaccines,
That's what I said. They don't tell anybody. Even there is a vaccine in there, I have no idea. If they give the Russians give a bottle, if there's a vaccine or something else, who knows? They didn't release any information to the world, especially the scientific world. Okay, I agree. I understand the confidentiality or patent rights or IP issues and blah blah blah. But in the scientific publications, they have to publish all the vaccine candidates that I mentioned in my talk. They are published. They are online, available from the journals for free of cost. If anybody of you just go there online from various eminent publications, you can read them. And that is like, with multiple clinical trials, multiple candidates, data from multiple doses, multiple number of experiments available. So that is what is the reliability. So Russian vaccine, what is in there? I know as much as you know. That's all. From these news channels and newspapers, other things. That's all.
Those who are in Zoom platform, they can unmute and ask questions.
I think administrator has to unmute everybody, because people cannot unmute themselves. Yes. It's controlled, I guess. Yes.
Unmute all.
You're breaking up. Can you repeat the question, please? Yes.
Hello.
Yes, please go ahead.
Sir, this is Sadhanandam, research scholar from Sathavahana University.
Hello, how are you?
Sir, good, sir. Sir, how much time to take to produce a oral vaccine, sir?
So oral vaccine, right?
Yeah.
Yeah.
Like polio drops.
I know.
Polio drops.
Years and years and years. First, we need to develop a potential vaccine that can be administered either intravenously or dermally. Then, once you have a candidate, then we start modifying it to make it more orally available. Vaccine, when it was developed, it was intravenously administered, and after like many years, 20-30 years later, orally available polio vaccine came into the so many years. To answer your question, many years. First, we need a vaccine.
Thank you, sir.
Thank you very much, sir. Due to lack of time, there are many questions, but due to lack of time, we are unable to discuss with you.
Anybody has any questions, they can reach out to me in Twitter or Facebook, and I'll be happy to answer.